Hereby, I open this academic ceremony in which David Diouf will defend the academic thesis, Balancing the Brain, Precision Gene Editing in Scalable Cultures of Human Stem Cell-Derived Neural Circuits, the case for ST3GAL3. First, I will also introduce the promotion team, who is opposite from me. The first supervisor is Professor Van den Hove, holding a chair in neuroepigenetics at Maastricht University. Then there is the second promoter, Professor Klaus-Peter Lesch, with a chair in molecular psychiatry, Center of Mental Health in the University Hospital Würzburg in Germany, and the co-supervisor, Dr. Tim van Mierlo, with expertise area repair-inducing cognition enhancers, also from Maastricht University. And now I may invite you to present a summary of your study and the conclusions of your thesis.
Dear Pro-Rector, honorable members of the Corona family, friends, and colleagues, thank you for being here. I'm happy to finally stand in front of you to present the research I've conducted during my PhD years and that led to the finalization of my thesis, entitled Balancing the Brain, Precision Gene Editing in Scalable Cultures of Human Stem Cell-Derived Neural Circuits, the case for ST3GAL3. In the upcoming 15 minutes, I'll present to you my main findings, starting from a simple yet elegant concept: the excitatory-inhibitory brain balance. As many of you know, our brain is formed of billions of specialized cells called neurons. These neurons are constantly communicating with one another using tiny pulses of electricity. When one neuron sends a signal to the next one, it often acts like a go signal, exciting the second neuron to propagate the electricity. Other neurons instead do the exact opposite. They send stop signals, inhibiting the electrical pulses of the next neurons. To understand why this matters, I want you to imagine a symphony orchestra. For the music to be beautiful, every musician must play exactly at the right volume. If the brass section is too loud, they drown the violins. But if the percussion is too quiet, the whole orchestra can lose the rhythm. The balanced dynamic shifts in the dynamics from each musician are what lead to the sophistication and variety of music. Our brain is just like an orchestra, but formed by its billions of players, the neurons, and it's through the dynamic shifts in excitatory and inhibitory activity that it can perform its most sophisticated functions. When the balance is just right, we can learn, focus, and remember. But when the balance is lost, the harmony can turn into noise. This noise, the disruption of the excitatory-inhibitory balance, is a hallmark of a group of conditions that we refer to as neurodevelopmental disorders. These include, among others, autism, intellectual disability, ADHD, and epilepsy. We often hear the term spectrum in relation to these disorders. This is because subjects with different entities often share similar core challenges. These overlap often blurs the lines in between different entities, making it hard to understand when one ends and the other one begins. What is fascinating and what my research actually focuses on is that this overlap isn't just seen in behavior, it is also present in the underlying neurobiological struggle. These seemingly different conditions all seem to be struggling with the same root problem, which is a noisy brain because the excitatory-inhibitory balance has been disrupted. For decades, we've known that NDDs tend to run in families, are heritable. If a parent has one of these conditions, the children are much more likely to develop them as well. This strong family link tells us that the origin of NDDs is partially in our DNA. Because genetics plays such a crucial dominant role, it becomes extremely important in order to identify the genetic conductors behind the scenes, namely those specific genes that are responsible for governing the E-I balance of the brain. My research has been a quest into identifying one of these genes. Its name is ST3GAL3. And my goal was to understand how this single gene can conduct the whole brain orchestra, and how this gene, when altered, can throw the entire brain's orchestra out of tune, leading to the development of NDDs. But what made me suspect that ST3 indeed acts as a primary conductor of the EI balance? The answer starts with a clue provided by nature. There are about 23 patients in the world with a rare severe mutation in ST3. These patients suffer from severe intellectual disability and epilepsy. But the story doesn't end with these few patients. When previous research looked at the DNA of thousands of people in the general populations, they found that a subtle variation in this very same gene is also linked to ADHD. By linking more subtle neurodevelopmental disorders like ADHD to more severe conditions like intellectual disability and epilepsy, ST3 seems to be at the very heart of the NDD spectrum. Despite these associations, remarkably little, though, is known about the actual role of ST3 in the brain, and virtually nothing is known about its contribution to the excitatory-inhibitory balance. This gap in knowledge is what pushed me into investigate. Is ST3 indeed a master conductor of the EI balance on the brain? And is ST3 and its genetic alterations responsible for the disruption of this balance, leading to NDDs? Because if we can understand how this simple gene works in our brain and perhaps learn how to manipulate it, we might be able to help not just 23 patients, but millions of people in the world dealing with neurodevelopmental disorders. But so how do we study the function of a specific gene in the human brain? We can't exactly take a piece of a living person's brain to experiment on, and working with animals, we would lose the human DNA. Hence, we would lose the human genetic complexity that we specifically aim to understand better. This is where the revolutionary role of stem cells come into place. And specifically, we made use of a technique called induced pluripotent stem cells. We take a simple skin sample from a human, and we can keep it alive growing it in a dish. Therefore, by exposing these skin cells to the right set of molecules, through a process that actually won the Nobel Prize, we can reprogram these cells back into stem cells, called iPSCs. This initial reprogramming is crucial. Indeed, stem cells have an immense potential, which is held within their capacity to differentiate into any cell type of our body, including our brain. So you might see where this is going. By exposing our iPSCs to the right set of molecules, we can transform them into living human neurons in a dish, so that we can investigate the human brain without having to open anybody's skull. But then, how do we specifically investigate the role of ST3 within our neurons in a dish? To see if indeed ST3 was crucial for the proper functioning of our neurons, we needed to work with two lines, which would be identical except for this specific gene. One healthy, normal line, and one line where we completely and only eliminate the ST3, what we refer to as the knockout. Therefore, by comparing the neurons from the healthy line to the neurons without the ST3 gene, we could see if indeed the absence of the gene would lead to changes in our neural circuits. Therefore, the very first part of my work in the laboratory focused into generating this ST3 knockout stem cell line. I made use of molecular scissors called CRISPR-Cas9. What makes these scissors particularly powerful is that they can be programmed to actually find one specific gene. I directed them, of course, to hunt for the ST3 gene. We therefore introduced these molecular scissors into a healthy stem cell line we had previously generated, and they precisely knocked out our ST3 gene. But as with any complex surgery, we couldn't just assume that everything worked perfectly, so we had to perform a series of checks in order to detect that everything went according to plan. Indeed, when we were reading the DNA of our 2 lines, we could confirm that SCALABLE was completely eliminated in our SCALABLE knockout line, whereas was perfectly intact in our healthy line. Additionally, we could also confirm that our generated SCALABLE knockout line also maintained the stem cell potential, as you can observe and appreciate in this image by the similar levels of green fluorescence in both lines. At this point, we had in our hands the ideal experimental setup. 2 sets of human stem cell lines, which were identical in every single way, except for the presence or absence of the SCALABLE gene, and that we could transform into neurons. This would allow us to say with certainty that any change that we were detecting in their behavior were indeed given by the presence or the absence of that gene. Therefore, at this point, we only needed to transform them into neurons, but it is a process that is easier said than done. In our field, there are generally 2 different ways or protocols in order to transform stem cells into neurons. Each method has its own recipe of molecules. And to make sure that our results weren't just a side effect of the specific recipe we were going to choose, I decided to make use of both of them. We can simply refer to them as Protocol A and Protocol B. I wanted to see if indeed the changes that we predicted were showing up regardless of the protocol that we adopted. As you can see in these images on the slide, both protocols were successful, leading to the generation of beautiful, complex human neuron networks in a dish. This would give us double the confidence that any findings that we would find at this point were indeed given by the gene itself and not by the way in which we obtained our neurons. So at the point that we had the neurons in a dish in our hands, the very first thing that I wanted to look at was the blueprint, namely their molecular composition. I wanted to see if indeed, even after removing SCALABLE, our neural circuits still had all the right molecules to build their connections. And we found something startling. When you remove SCALABLE, the cell essentially forgets how to build neuron-to-neuron connections. Our SCALABLE knockout line presented a massive decrease in the molecules responsible for building these essential connections. And importantly, a specific set of molecules particularly responsible for the formation of inhibitory connections were the most severely affected. This led me to think that indeed, if our neurons do no longer have the right tools in order to build the connections among themselves, they might no longer be able to communicate properly with their electrical pulses, hence leading to disruptions in the EI balance. At this point, therefore, I decided to look at their electrical activity, at the electrical activity of our neurons in a dish. And I wanted to see if the absence of SCALABLE actually led to changes in the way how our neurons communicate. I therefore needed to listen to them. For this purpose, I grew our neural circuits onto microchips. These microchips allow us to record the electrical activity of neurons in real time. So what did we hear? Just as the blueprint suggested, the healthy orchestra played in organized rhythmic patterns. They knew when to start and they knew when to stop. But in the circuits without SCALABLE, where the blueprints for the neurons' connections were lost, we found that the pattern was much more chaotic. The neurons were hyper-excitable, and they fired with irregularly, massively longer bursts of electricity. In other words, the overall balance of the orchestra was lost, leading to noise. So what could I finally conclude? My research has shown that indeed SCALABLE is a master conductor of the brain's orchestra. We have demonstrated that its function is indeed essential for the proper maintenance of the delicate and dynamic excitatory inhibitory balance of our neurons. We also shown that alteration by removing this gene, the entire system loses its functional stability, resulting in the chaotic patterns of electrical firing that we observed in our microchips. Furthermore, by connecting the electrical findings to the molecular findings that we obtained, we were able to pinpoint the specific role of SCALABLE into building these neuron-to-neuron connections. with a vital impact on inhibitory ones. Our molecular analysis also allowed us to identify the whole support crew, namely all the other molecules that are indeed working together with SCALABLE in order to perform its tasks. So what does this actually entangle for the future? This research has transformed what was initially recognized as a rare mutation in a subset of patients into a set of quantifiable and druggable targets. These findings represent the very first and crucial step in order to identify new treatments that could alter the course of neurodevelopmental disorders. By modulating the SCALABLE gene, indeed, or its molecular partners, we might be able to restore the excitatory-inhibitory balance in patients, finally moving beyond managing symptoms and beginning to restore the fundamental harmony of the human brain. With this, I would like to thank you and give the word back to the Prorector.
Thank you for this nice presentation. The opposition will now be opened by Professor Harwood. He's chair of the assessment committee and also secretary, holding a chair in integrative neuropsychiatric cell biology at the Maastricht University. The floor is yours.
Thank you. Thank you, Prorector. And thank you, dear candidate, for actually a very clear talk that simplifies a very complicated field. So well done. I thought I'd like to start really at a higher level of thinking about this problem, and I thought I would start with Proposition 7. So if somebody don't mind reading that out.
While the definition of behavioral dysfunction is ultimately culturally determined, the identification of objective biological markers transcends these subjective thresholds to provide a universal basis for diagnosis.
Well done. That's quite a long statement, but quite an interesting one as well. Okay, so I would like to contest that. And I'd like to contest that by arguing that behavior is a biological marker, just as much as anyone else. So I thought we could have a discussion about this, because it sets up the sort of premise of your work. So just in your own thoughts, how would you define behavioral dysfunction, which is clearly something that you recognize as subjective?
So highly esteemed opponent, thank you very much, first of all, for your kind words and for your question. First of all, you're asking the, what would I define as disruptive behavior? When it comes to neurodevelopmental disorders and the definition of their behavior and the way how diagnosis goes, it's indeed, as we all know, described in the DSM-5. Over there, they have a whole cluster of symptoms for which they always assess, according to which one you present, which entity theoretically you would fall into. But indeed, when it comes to entities, we see that there is this massive overlap among them. So it becomes already trickier to identify which one specifically you're developing in that sense. When it comes to actually defining what is a disruptive behavior, indeed, what the statement refers to is that indeed that is very much cultural dependent and subject dependent. So what is indeed perceived in certain populations, for example, in certain cultural background as being disruptive, in other populations might not be perceived as such, or at least not to the same level of extent. This different perception in terms of how much disruptive indeed a certain behavior is leads to the differential experience that a different subject might have in those different cultures, and therefore also the differences in which the specific subjects might go after, might go after specific treatments or go to visit a doctor, so to specialized opinions.
Okay, and so how would you define a biological marker then?
Yeah. So as you mentioned, indeed, behavior can absolutely be considered as a biological marker, and I would definitely agree with you in that sense. What this statement indeed specifically was referring to, and what I'm happy that indeed stimulated the conversation about, is that indeed when it comes to behavior, it is indeed a biological marker, but it, as I just mentioned, very subjective, and depending on the different populations that we're talking about, or even, for instance, like when it comes to sex differences in the way how they manifest those symptoms and stuff. Whereas if we talk about, for instance, instead genetic screening of our subjects, then in that case, our perspective loses the bias of the subjectiveness and becomes more empirical and based on the actual data that we have.
Okay, great. So do you think it's possible then to have a truly, especially based on your experience, a truly objective biological marker, or will there always be subjectivity and objectivity?
Well, if we talk about the individual marker, I believe that indeed one of them can be purely objective. So if indeed we start screening everybody, let's say everybody that goes into a clinic in order to address their issues, and we screen them genetically speaking, those specific set of screenings, those would be, of course, in themselves objective. Every time, of course, up until this point, especially, like, the majority of entities are still mostly diagnosed based on behavioral symptoms. So even when combining these genetic data together with the behavioral that we would assess, then of course the subjectiveness would come into play again. But by using genetics or any actually different molecular sets of assessment that we could have, we would at least, let's say, be more certain, like allow the clinicians and give the clinicians with better tools, more objective tools that would allow them to actually be more precise about that, and also probably perhaps also understand better the individual, because one thing that I truly advocate also in my thesis is moving beyond this categorization of entities, but the idea of a more spectrum, a more divergent manifestation of the symptoms. So if indeed we start considering the patients not just based on you have this entity or this other entity, we might move more towards what actually is going wrong with you, or you feel is going wrong, and then we can work on the issue in itself rather than on the manifestation of it.
I'm okay for a follow-up question, yeah? Okay, so that leads us to the gene that you're interested in. So do you think that will give you a biological marker, or maybe a good diagnostic tool, or do you think it needs to be, how would you use it?
Yeah, I certainly believe that indeed it is in itself a very interesting biological marker that we could use in the clinics. I do believe that indeed this kind of research, like I am passionate about, in the sense that I would love to see even more, in the sense of like also investigating different kinds of genes, of course, that I am completely oblivious of. And then by, of course, starting to build, for instance, a set of different kind of genes that might be involved, or we find to be involved, for instance, in the EI balance, like we saw for S3, we might therefore be able to be more certain about that and have a more complete set of checks, let's say.
Okay, great. So you think it will contribute? Yeah.
Absolutely, yeah, I do. Yeah.
Okay, now the opposition will be continued by Professor Homberg, present online, also member of the assessment committee, having a chair in behavioral neurogenetics at Radboud University Medical Center in Nijmegen. And you can see her over there.
Dear candidate, first of all, I would like to congratulate you with your thesis. I have read it with pleasure, and although I'm not an expert in cell models, I think you presented your work in such a way that also non-experts can understand it, and it was also very clear during your layman talk. So that is, I really appreciate this, and of course also very nice for all the people who are in person during your thesis defense. And I also think that the topic of your thesis is both scientifically and also societally very, very relevant. So after reading your thesis, some questions came up into my mind. So as I mentioned, I'm not an expert in cell models, but I was thinking a little bit bigger in terms of whole body processes. And so what I learned in the past is that CELL-DERIVED, which is the target of the mutation. So you speak it out very fast, but I'm a little bit slower. The ST3GAL3 mutation is transferring CELL to NCAM1. And an important source of the CELL is actually the breast milk. So the breast milk contains human milk oligosaccharides. Especially, there are two types. the 3-sialyl lactose and the 6-sialyl lactose in the mother milk, which transfer the sialyl to from the breast milk to the baby. So I was actually wondering what would happen if children are fed formula milk. So instead of receiving the breast milk. So the formula milk does not contain any of these sialyl lactoses. So would that be, how would that affect the children with the mutation? Would formula-fed children also be negatively affected in their brain development, or vice versa? Would addition of these human milk oligosaccharides help to develop the children slightly better? So, what are your thoughts about this?
Yeah. So, highly esteemed opponent, first of all, thank you very much again for your kind words and for your question. Indeed, you brought up one of the concepts that I believe, that I found to be the most interesting for myself as well. So I talk about all the time about these genetic mutations, or either subtle or like more severe, leading to indeed these different kinds of NDDs. But indeed, you also mentioned that sialic acid, which is the main substrate that S3 uses, and transfer it actually mostly in our brain onto proteins, like on glycoproteins, it's of course of extreme interest. So as you mentioned, it is present in maternal milk, and it is not present indeed in formulates. And there is already studies out there showing that indeed there is a correlation between children that have not been lactated maternally and the development of NDDs. Now, is this a direct consequence of the presence or absence of sialic acids in the formulate compared to the maternal milk? Well, it's a bit of a strong statement at this point to immediately infer that, but of course the indication seems to be, or at least this information seems to be indicating towards that. So in my opinion, for instance, if we think about treatment of different subjects with NDDs, or even, for example, just general information of the population, it might be extremely important in order to stress that we could just supplement more sialic acid, especially, for example, in formula milk, in order to prevent the development of NDDs. Or perhaps we could even test, of course, in patients that already present different NDDs, the supplementation with sialic acid, where that would already ameliorate at least the symptoms. Now, of course, when we're dealing with neurodevelopmental disorders, that we're dealing with the continuous process, which therefore, not in all cases, I believe, that might be fruitful. So if we are intervening too late by giving sialic acid and the neurodevelopmental processes have already been taking place, perhaps that might not solve the issue. But indeed, I do strongly believe that I am confident that the substrate and not simply the gene and the related protein might be the right target, but the substrate as well.
Okay, thank you very much. So then, based on your answer, I had another question that came into my mind. So at what stage during development, so for instance already embryonic development, will the mutation that you studied have an effect on brain development? Is that already known? Is it starting already when the baby is an embryo, or what stage already does it start to influence brain development? Yeah,
that's an extremely valid question. So at this point, we still don't know exactly, indeed, when this mutation plays its role. It is very much more likely, though, that this mutation actually plays its role, or like even more subtle mutations. We can even think about, for instance, epigenetics in that sense, that might silence the gene in the, rather than completely suppressing it, might already start having an impact from pretty much all phases, indeed, of our formation, like of our brain formation. So if we think about the processes that are in there, so from synaptogenesis to myelin formation to the inclusion of astrocytes into the synaptic clefts and their contribution into that, all of these processes are processes that do require a very high level of communication among the cells. Given the role of S3 that seems to be indeed impacting the cell-to-cell communication, I wouldn't be surprised if these already starts from the very first formation of synapses in the brain and all the way impacting even the formation of the myelin and, for instance, even the pruning, because that is then performed by microglia at that level. So we do not have one specific time frame in which SCALABLE seems to be playing a role, but very likely, in my opinion, it actually will impact all of it pretty much.
Okay, thank you. And just out of curiosity, so what is the age of the cell lines that you used? Does it have an age? in
Absolutely. So when it comes to working with iPSC-derived neurons, indeed, there is always the question of whether, indeed, we're talking about mature neurons or are there more juvenile neurons. The neurons that I was using, according, let's say, to the protocols and to the assessments that we perform, those are considered to be mature neurons. What does that mean for human age? I would not be able to give you a specific number, let's say. We're still working with, indeed, neurons that are just present in a dish. There is, of course, a series of checks that we perform and that the previous laboratories that establish those protocols perform in order to assess that indeed they behave the same way as, for instance, mature cortical neurons from developed individuals would behave. So these are indeed day 30 for protocol A and day 49, if I remember correctly, for protocol B, but I cannot truly give you an age, let's say, that they would correspond to.
Okay, thank you. I was also curious whether your cell lines have a sex. So and whether sex could interfere or interact with the mutation.
Yeah, absolutely. So there is also one thing that we really also see in patients. So patients with different sexes get diagnosed differently already with different NDDs. So very likely indeed there is a component in there, although it could also, like part of it can be explained by diagnostic biases, of course. Our cell line indeed has a sex, so it has been derived from a female individual. These can always have an impact. One thing that, for instance, can impact on is, genetically speaking, of course, the presence, or possible reactivation of the X chromosome that indeed previous studies have shown that might happen in iPSCs. It seemed indeed not to be the case in our iPSC line when we performed also the karyotype assessment. So indeed, at that level, the different genetic background that we use might always have an impact, and that's the reason why I always stress, and I particularly stressed even in my experiments, the use of an isogenic line, so that we would truly be able to pinpoint whether S3 actually plays a role by using the exact same genetic background when looking eventually at the findings. Because indeed, if we wanted to include, for instance, different genetic backgrounds from a male line, for instance, that would also require, though, a much larger sample size, right? Because we wouldn't be able to just include one line. That would be like comparing one person to another completely different person and then just assume any findings based on that. So we would need to include, let's say, 10 lines from females and 10 lines from males, and that, of course, would basically change my whole project at that level. But I do believe that it's also extremely important to proceed in that sense, so really using subject-derived lines, iPSC lines. Yeah.
Do I still have time, or is it
No,
time?
no, I'm sorry. We would like to continue, and the opposition will now be continued with Professor Grunblood, holding a chair in translational molecular psychiatry of the University of Zurich, Switzerland. Thank you for joining, and the floor is yours.
Professor Bloom is online. This is not
candidate. You presented very nicely the influence of ST3-GAL3 gene in neurodevelopmental disorders, as well as the epilepsy. And these are two mainly monogenetic disorders, as well as showing that there are some associations in ADHD which are rather polygenetic. Now, coming to evidence at the clinical level, I was wondering if there is any evidence shown regarding the EI orchestration that you have mentioned with this gene in clinical aspects for ADHD, for example. And if you can elaborate a little bit more how this might be affected in this clinical group.
Yeah, highly esteemed opponent. Thank you very much for your question. So when it comes to ST3-GAL3, I do not, I cannot recall, but I'm quite certain indeed there is not truly a one paper out, let's say, that was looking clinically at all the patients with ADHD and presenting the subtotal variation in S3 comparing it with the population. As far as my knowledge goes, of course, I don't think there is any papers out there clinically looking at that difference. When it comes, though, to the EI balance and the effect, and its foundation on different entities, for instance, ADHD in that sense, so for more polygenic entities, there is already actually quite a lot of evidence out there that seems to be showing that indeed that seems to be one of the root problems, let's say, for ADHD. So, for instance, we see that there's some fMRI studies that looked at the population, both juvenile or adult populations, and looking at the presence of GABA in the brain and just overall GABA activity in the brain, and they were seeing that indeed there seems to be quite a massive reduction in the juvenile phase, which is instead reversed in the adult phase. Now, it's very speculative to argue about, it's very tempting to speculate that this is the reason why, for instance, in ADHD, we see that the NDD seems to be fading over time, and it might therefore be a compensatory mechanism from our brain in order to reestablish the EI balance. And when that is successful, indeed, the ADHD goes away. But it's indeed, once more, a speculation that we still are doing at this point. It's not truly causative.
Good. So that brings me a little bit to the thoughts. So as you mentioned that there is some evidence of reduction in GABA, at least in the juvenile phase. And as much as I know, there are also evidence of increased glutamate in ADHD. Yeah. How would you translate it into clinical phenotyping that you might see in the ADHD and the functionality that is in a way then linked to this EI imbalance?
Absolutely.
What type of behavior would it correlate?
Yeah, indeed. One of the reasons why I particularly like, let's say, the EI balance and the way how it falls within the NDD spectrum. As I mentioned also in my presentation, indeed, we are not talking about one specific disorder. We are talking about many different ones that have a lot of overlap. So, of course, we cannot assume that all of them have exactly the same issue at the back. But the EI balance in that sense falls very nicely into creating, let's say, a framework in which we could conceptualize entities. So, for instance, if we think about disruption of the EI balance, we're not talking about just a static like overexpression of glutamate, for instance, compared to GABA. We're talking about a process that is highly dynamic. So, and we're talking about both the spatial scale, so different brain regions, for instance, that might be affected, but we're also talking about the temporal scale. So are we actually talking about the oscillations patterns, for instance, that are fundamental for our hippocampal functioning? Are we talking about like more tonic firing? So like the time frame in which we truly want to observe that and the spatial scale into which we can see those disruptions might fall into the formation of different NDDs.
Okay. Moving to a different type of question, you didn't mention that specifically.
I have to interrupt you. A short question is allowed now and a short answer. We would like also the other opponents to have some time.
I'll try to make it short. So you used the bulk RNA sequencing in your experiments. Could you just mention very shortly the pros and cons and how you could solve it?
Absolutely. So pros and cons of bulk RNA sequencing in these cases indeed that we are taking the whole neurons that we have in our dish and then looking at the transcriptomics of it. More advanced techniques such as single-cell RNA sequencing might actually give us a really good insight into, for instance, identifying cell-specific alterations of their RNA transcripts. The bulk RNA, though, in that sense, also give us a very clear understanding, for instance, with such a strong differential expression into what are the different processes that might be affected over there. So indeed, like in my dream, like in the experiments of my dream, I would include also single cell RNA sequencing, let's say, for the future. Yeah.
Thank you.
Thank you. And now the opposition will be continued with Dr. Scapus. She's present on site here, having expertise in behavioral and molecular neuroscience at Maastricht University.
Thank you, Prorector. Dear candidate, first of all, let me start with congratulating you on achieving this beautiful thesis. Completing a PhD is already an achievement, but doing it in a joint doctoral degree, working in different universities, different countries, is an additional achievement as it requires a bit extra adaptability. So very well done. I would also like to congratulate the promotion team because this project, I believe, very nicely brings together expertise in neuroscience, genetics, stem cell biology, and renders this very nice result we saw today. I read your thesis with great interest, but of course, I also would like to raise a question, and I would like to kick off with a methodological choice. Because in chapter 3, you mentioned that with the directed differentiation, you often lack fully maturity of your neurons compared to the induced differentiation. Then you also discuss in your general discussion that the differences or similarities actually you see in your transcriptomic profiles are consistent over methodological choices you made, so protocol A, protocol B. However, when we look at the moment that you analyzed your neuronal cultures, we see in the directed differentiation that lacks maturity or is hampered or delays the maturity, you're taking earlier time point when you compare it to your induced differentiation. Because with your directed, you're taking 30 days in vitro. With your induced, you're taking 49 days. You also see in your PCA plot that your wild-type protocol A clusters a bit with your knockout protocol B. With that,
for your kind words, very much appreciated. And for your question, indeed, you're raising a very valid point. The two protocols, as I discussed also like in one of the chapters, have advantages, pros and cons. One of which, indeed, directed differentiation being often to the formation of immature neurons. So when, though, I talk about, for instance, the day 30 differentiated neurons with directed differentiation, it's important to understand that also these differentiation starts from the moment of plating of our NPCs, so neural precursor cells, right? So in order to differentiate our neurons, we plate our NPCs and we differentiate them for 30 days. That doesn't mean, let's say, that before that nothing happened, so we still had all the formation of the NPCs before. So although indeed in paper it might seem that, yeah, they are more juvenile, actually this whole process started much earlier on with the formation of embryoid bodies and neural rosettes and the selection of them that leads to then the formation of NPCs. So indeed the timing eventually becomes longer, much longer, of course, compared to the induced differentiation protocol that I'm using. When you refer to the transcriptomic data and the discrepancy that we see at that level, indeed, the directed differentiation protocol, one of the main aspects of it, which can be considered both as a pro or a cons, is the fact that it leads to a generation of let's say a heterogeneous population of neurons. So we cannot indeed control the specific subtypes, the specific cellular subtypes that we have in there. That discrepancy that we have in the two, let's say in the protocol A wild type especially, we have two groups that are segregating independently, might indeed be given by that. So of course, when you cannot control that specific homogeneity, it might be more likely that they segregate more independently from each other according to batch or different effects. But that, of course, was not picked up, so I still had to include them in the analysis. And that specific separated segregation was actually what probably led into the lower levels of differential expressed genes that we then eventually saw. Because, of course, the more you increase the within-group variability, the less likely you're going to be to also find between-group variability. So, yeah.
Thank you very much. Now moving to a bit more conceptual aspect. So on page 52, you briefly discussed gamma-late B6 as a GABAergic compound, which showed in 2020 some promising results. And I went a bit into a rabbit hole because the gamma-late B6 contains quite a substantial amount of vitamin B6, which is being phosphorylated to coenzyme important in sphingolipid metabolism. And sphingolipid metabolism is important in myelination. So maybe you start feeling it. Do you think promising effects seen with gamma-late B6 might be at least partially attributed to enhanced myelination? And can we actually restore the excitatory-inhibitory balance by just targeting glia cells instead of neurons?
Absolutely. So one thing that I briefly touched upon before is indeed, in my thesis, I specifically focused on GABAergic and glutamatergic cells, being them the main actors, let's say, when it comes to the EI balance. But many other cell types, of course, are in our brain, and especially one thing that seemed to be particularly important also for the EI brain, of course, is the myelination. Indeed, when it comes to SCALABLE, it's a bit of a conundrum, indeed, that I found myself into as well, because indeed SCALABLE seems to be highly specific actually for glycoproteins in the brain. So probably, indeed, is not SCALABLE in itself, but other members of the family that seems to be more specific instead for lipids, and then the lipid metabolism being then highly important, of course, for myelination processes. Although, if you think about it, then eventually in that sense, for instance, GABAergic interneurons, parvalbumin positive interneurons, are highly myelinated. And those seem to be indeed the ones that are the most affected, for instance, in ADHD, in our hippocampus and stuff. So I truly believe that indeed by, at least partially, working with myelination and oligodendrocytes might be able to restore, or at least help ameliorate the EI balance in neurodevelopmental disorders, absolutely.
I couldn't agree more. Then maybe a final, shorter question. You're talking about the spectrum, right? But in NDDs. But you are studying a complete loss of function of your ST3 GAL3. Do you think your model is rather modeling something on the very extreme ends, or do you think it can be used for other NDD disorders, especially when thinking about rescuing studies?
Yeah. So indeed, I am using, let's say, actually probably what you can consider as the most extreme model in that sense. The reason why I opted for that first is that because at the moment, indeed, as I presented in my presentation, there is almost nothing known about the exact functioning of SCALABLE in the brain. So I needed to know which processes would be even altered or impacted at all by the presence or absence of SCALABLE. When it comes to modeling then polygenic disorders in that sense, SCALABLE, though, it's still important, and our model can still be valid in the sense that we, for instance, can rescue indeed, and then see the causative effects of SCALABLE in those specific processes. But also other effects that we can try to aim for are, for instance, hypermethylation of the gene, to see if indeed we can have a more gradient results of the SCALABLE. So definitely, I can think of like many different models, from microRNAs to the use of epigenetic editing tools in which we could actually upregulate or downregulate SCALABLE rather than knock it out, to have a better insight into its functioning in the brain.
Thank you.
Okay, now the opposition will be continued by Professor Haaf, present online, having a chair in human genetics, and he is from the Biocenter of Hoopland, Würzburg University in Germany.
Dear candidate, also congratulations from my side. I want to emphasize a bit that your favorite gene, ST3-GAL3, was the top hit in GBOS studies. And usually these variants are of very small effect size. On the other hand, we have exome sequencing, genome sequencing, and indeed there's a growing number of pathogenic mutations identified causing different monogenic diseases. Can you expand a bit on what are the advantages of both, the disadvantages of GWAS versus sequencing, genome sequencing?
Yeah, highly esteemed opponent, thank you very much for your words and your question. Would you mind if I ask you again to rephrase your question? Which two models would I need to elaborate on?
Not models, approaches to get interesting genes. For example, you can use GWAS studies and you can use sequencing studies. What would you prefer, and what are the advantages, disadvantages?
Absolutely. So indeed, it's a very interesting point. I presented in my data also how much SCALABLE seems to be both a rare mutation that leads to a rare neurodevelopmental disorders, but also associated with polygenic disorders. This is indeed not so much of a dichotomy. So if we're doing, for instance, GWAS studies, then we will be able to pick up much more those rare mutations that are related to rare neurodevelopmental disorders. So for instance, those 23 patients with the mutation in SCALABLE. Whereas if we're looking, for instance, at EWAS studies, then perhaps we might be able to identify better polygenic disorders in which different genes and their epigenetic regulation might be leading or the cause or like at least at the root of the pathology investigated.
And is there any genotype-phenotype correlation? For example, what distinguishes a SNP in GWAS from the pathogenic mutations?
Yeah, indeed. There is also like a different SNP often can lead indeed to different epigenetic regulation of a specific gene. So when it comes to that, the combination of the two, of course, will be necessary. So the use of multi-omics in that sense and different computational analysis, it's always going to give a better insight into actually what is happening at the genomic level or epigenomic level.
As you have mentioned, most NDDs are actually of multifactorial origin, meaning you have different genetic factors and environmental factors. Maybe you can tell us a bit about the environmental factors. One we already had, the breastfeeding or not. What environmental factors influence the NDD risk?
Absolutely. So one thing that I briefly touched upon also in my presentation is, for instance, the heritability that we see. So family indeed plays a crucial role into that sense, and not just with their DNA, but also to the kind of environments that they foster. So a more stressful situation in the family can indeed already lead to different entities. Even before that, like a stressful gestation, for instance, from the mother during birth, before birth, also is associated with the development of different entities. So indeed, our brain is constantly, assimilating different stimuli from the external world, and the way how these then indeed impact the EI balance is more unpredictable. But then when combined with the genetic level, we can have these gene-environment interactions that lead or might be explanatory for different entities.
If you have a genetic factor, it's usually present throughout life. With the environmental factors, you have different exposure times. What do you think is more harmful, exposure at early development or later in life?
Certainly enough, when we're talking about neurodevelopmental disorders, I would definitely refer to early on environmental stressful impact, stressors having a higher impact. The reason is because, of course, our brain is still forming almost all the way up until 20 years old when we have the latest phases of synaptic pruning. So any potential external or internal impact into these very delicate phases of brain formation might have a stronger impact compared to having stressful that are indeed coming eventually later on when our brain is more stabilized in that sense.
Maybe a final technical question. If you compared the transcriptomes of your knockout lines versus controls, almost all differentially methylated genes were downregulated in the knockout. Can you comment on this? Why is that, and what is a possible mechanism?
Absolutely. So indeed, when we look at the specific gene sets, like so the different gene ontology terms that we were identifying, they were all mostly downregulated. That doesn't mean that indeed all of the genes were downregulated. When we look at the volcano plots, indeed, we also see that there is a lot of genes that seem to be upregulated, but seem not to be popping up in the gene ontology analysis. One of the reasons why I do believe that, for instance, when we're looking about synaptic related genes seem to be downregulated, I believe it's because of, I'm confident it might be actually because of different compensatory mechanisms. So we are eliminating the gene SCALABLE, which is not a transcription factor. It actually acts as a translation modification protein. So this is probably the effects that we see then at the transcriptomic level is probably a more secondary effect, so a more like response from the cell to something that is already happening because of the absence of S3. And in my opinion, it might be related to the absence, of course, of sialic epitopes on the cell surface, which leads to a higher proximity of the cells. And therefore, perhaps we have this compensatory mechanism, the cells trying to decrease at least the expression of synaptic related markers in order to decrease to some extent, to modulate to some extent these overly abundant connections that are starting to form.
Thank you.
Thank you.
Okay, thank you. And now, last but not least, the opposition will be continued by Dr. Mulder, having expertise in lipid metabolism from the Department of Internal Medicine of the Erasmus University Medical Center in Rotterdam.
Thank you. First, congratulations to you and your team with a heavy thesis. Was a pleasure to read. So my expertise is lipids, so I tried to get to lipids. But first, really interesting. You mentioned that neurodevelopmental disorders are a spectrum from functional to dysfunctional. And I would like to argue or to ask you if you think that Alzheimer's disease would fit in this spectrum. I will tell you why. Because there are a number of genes, about 10 risk genes overlapping for the risk for ASD and Alzheimer's disease. So a good part of Alzheimer's disease patients, could it be a neurodevelopmental disorder showing up late?
Esteemed opponent, thank you very much for your kind words and for your question. It's very fascinating because I'm slowly transitioning indeed into working more with Alzheimer's disease rather than neurodevelopmental disorders. I did actually come across also that idea that indeed Alzheimer's disease might not just be a neurodegenerative disorder, but a neurodevelopmental one. It is not, we cannot rule it out, and I actually do find it extremely fascinating and plausible indeed, that if we have a more fragile network building during our early life, that this fragility also holds over time, and then of course the occurrence of different stressor from genetic to environmental over time, and the accumulation of these stressors might lead eventually to development of NDDs. And that's why then eventually you would see the overlap in genetic vulnerability.
Would be an idea too. Maybe it has already been studied, I don't know. But then I found something interesting because I found that ASD individuals are hypocholesтерolemic. They have low cholesterol in general, or part, low LDL, low lipoproteins, and you would wonder why this could be of interest. There's a Nature Medicine paper from 2023 showing alterations in the lipidome in ASD individuals in the periphery linked to LDL. There was a child identified with LDL receptor deletion. And then I found, you wonder what the link is, with your ST3-GAL3, that sialylates the glycans on the LDL receptor and on LDL itself, causing differences in function.
Yeah.
So this could reflect changes in the brain. So you could maybe detect changes in the blood, important, giving information on the brain, as the LDL receptor is highly expressed in the brain. And mice with the dysfunctional receptor have cognitive problems. Mm-hmm. So in your chapter 5, you look at RNA-seq. Do you know by heart if the LDL receptor popped up in your knockout?
By heart, I'll be able to remember, yeah.
You don't know, okay. So it's expressed highly in the blood-brain barriers and in the oligodendrocytes, so myelin is changed, in astrocytes and in neurons. So could you speculate, or would you be interested in studying it?
Yeah, absolutely. Like these kind of concepts actually are something that I do find extremely fascinating. I would be interested into investigating more for sure. There is actually in our group somebody that already was working with stress, let's say like the stress in a dish, we used to call it. So like different models of iPSCs, exposing it to cholesterol and seeing the potential effects. So one thing that I elaborated briefly upon before, of course, is that early stressors in life, environmental stressors, so for instance, that lead to higher levels of cholesterol indeed seem to have a correlation with the development of NDDs. So potentially creating a model in which we can also not just play around with our genes, but also with the cholesterol exposure might give us some...
It's not what I mean.
Okay.
It's not what because we don't know exactly what the LDL receptor is doing. I was thinking if the lipidome in the blood is changed, you could look in your cells at the lipidome, lipidomics, and if it's changed as well, you may have a peripheral marker to reflect changes in the brain.
Oh, absolutely.
That's what I was thinking of.
Absolutely. Yes, very
It's just a
fascinating.
weird idea, but...
It sounds like a great idea, absolutely. It is indeed very much possible, absolutely. Yeah.
And you are working on combining cells now, other cell types?
Yeah, so indeed, like at the moment, I'm actually also moving a little bit into other disorders, like I mentioned before, like for instance in Alzheimer. But indeed, like my idea over like these kind of projects is indeed to use a bottom-up system, so where we constantly add different cell types so that we can constantly be precise about what are the effects of the genetic mutation, in our case, for instance, in different cell types and how they eventually all lead to a more complex system and how these more complex systems function. So astrocytes, for instance, are extremely interesting for me. Oligodendrocytes, extremely interesting for me as well. Microglia, extremely interesting for me. But of course, it becomes more and more complicated then.
Oh, thank you very much.
David Diouf, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room. Okay. David Diouf, the degree committee here present has assessed the quality of your thesis and of your defense. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor van den Hove is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, David Diouf, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university.
You are invited to give the laudatio. Professor Lesch.
Director, dear members of the Corona, colleagues, family, friends, dear David. It is a great pleasure and honor to celebrate with you your successful defense today. The moment marks the culmination of a remarkable scientific, personal and truly European journey that has brought together institutions, disciplines and people across borders in the best possible way. David's path into neuroscience is already a story of curiosity and courage. After completing his bachelor degree in Parma, Italy, he went to Maastricht University for his master's degree in neuroscience, a cultural leap from Bella Italia that he mastered with openness and resilience. These early stages already showed the international mindset and flexibility that would later become a hallmark of his PhD work. In winter 21, David joined my lab of molecular psychiatry in Würzburg. He first worked within the framework of an EU project called Eat to be Nice, referring possibly to maternal milk, and later within the international training network Serotonin and Beyond until summer 24. This program allowed him to return to Maastricht for a 6-month secondment, strengthening and renewing the long-standing collaboration between Würzburg and Maastricht that has already been thriving for more than 15 years. David's thesis is thus not only an individual achievement, but also a product of sustained, fruitful collaboration between two research environments. This collaboration continued the arrangement of a joint PhD program between the universities of Maastricht and Würzburg, under which David has now successfully graduated. His work exemplifies what such joint programs are meant to achieve: scientific excellence, mobility, and the training of young, aspiring researchers who can move confidently between different academic cultures and infrastructures. Scientifically, David's achievements are rooted in genuine curiosity, original thinking, and methodological rigor. One major milestone of his work was the replacement of murine astrocytes, with human iPSC-derived astrocytes as a feeding layer for the glutamatergic GABAergic co-culture system. This is a demanding and forward-looking approach, bringing his experimental models closer to human biology and opening new ways to study excitatory and inhibitory balance in neuronal networks. It is exactly this combination of conceptual innovation and painstaking technical work that characterizes a strong PhD thesis. Equally important, David has contributed to the social and collaborative life in my lab. During his time in Würzburg, he established an excellent rapport with the technical staff Gabi Ortega, Julia Merck, and Nicole Schraut, and as well as the project manager Judith Stiller, who either joined us online today or sent warm regards. Scientific work is always a team effort, and David's ability to integrate himself into our team, to communicate, to share, and to support has greatly appreciated. We should also remember the many shared experiences beyond the bench that shaped these years. There was the farewell party for postdoc Xingping Ku in December 21, the lab hiking day to Wertheim Fortress with the boat cruise on the River Main and the barbecue in my garden in July 22, the Serotonin Club meeting in Berlin in 23, and the IPS network meeting at the European College of Neuropsychopharmacology in Zurich in April 24. These occasions created friendships, inspired scientific ideas, and anchored David in an international community of researchers. A particularly beautiful aspect in David's story is how his scientific and musical lives interweave. David holds a bachelor degree in music from the Parma Conservatory and has actively performed in the Luigi Cherubini Youth Orchestra. It is therefore no coincidence that in his defense presentation he compared the excitatory-inhibitory interaction of neuronal networks to the organized interplay within symphony orchestras. This metaphor is not only elegant, it grows directly out of his biography and shows how deeply he understands both systems, neuronal networks and musical ensembles, as complex, finely tuned structures. One of the most memorable shared events in this context was attending a performance of Anton Bruckner's symphony together in Würzburg Cathedral in July 24, commemorating the 200th anniversary of Bruckner's birth. Experiencing this monumental music in such a space offered a powerful illustration of harmony emerging from contrast, of tension and resolution, much like the dynamic balance of excitation and inhibition that lies at the heart of David's research. David, today we celebrate your intellectual achievements, your perseverance through experimental setbacks, your growth as an independent science, and your unique way of bringing together science and art. You are now about to continue your career as a postdoctoral researcher in Maastricht, and there is every reason to be confident that this environment will value and foster your talents. May your professional and personal future continue to unfold like a well-crafted symphony, with moments of bold excitation, phases of calm inhibition, and above all, a lasting balance that allows your ideas to resonate and inspire others. Congratulations, Dr. David Diouf, and all the very best for the road ahead. Thank you.
I will continue. We had 6 minutes each, you said. Oh, no. I will try to speed up.
I didn't say that.
No. I know you didn't. David, where to start? I will speed up, as I said. Well, you joined the Research Master, as Professor Lesch outlined, in 2018 that was, and I met you first in the psychiatric neuroscience course that I back then coordinated with Gunther. Tim met you in the same period. He met you in another course that Gunther was coordinating at the time on genetics. And Tim was teaching a brand new and back then rather futuristic technique called CRISPR-Cas that you also spoke about today. And as part of one of the assignments there, students, including you, had to design their own guide RNA. And you chose BDNF, brain-derived neurotrophic factor, back in 2019. And Tim still remembers that it sort of sparked something clearly with you, in a sense that after the lecture you actually went to his office and continued the discussion. And that's usually a promising sign, not always, but in this case it was. A bit later, well actually a bit later than you, we came across again, in the sense that our paths crossed. You went to Sun City in Arizona to Diego Mastroeni for your research master internship, which was a former PhD student of mine. And so I was your institutional supervisor on behalf of Maastricht University back then. And then you went to Würzburg, as Professor Lesch nicely outlined, which also at a certain moment had me on board in their supervision team. And then Tim again came along, because in the end you moved from Würzburg to Maastricht, and that secondment took a bit longer in the end. And you started working both in Maastricht but also in Hasselt in Tim's lab over there. And interestingly there in 2023, in that same genetic course that initially Tim was lecturing in on BDNF, this BDNF gene retired gracefully as the example gene and now was replaced by ST3-GAL3 based on your first author publication. In the meantime, you actually took over the lecture, and soon we all will be replaced by you in many ways, and you are definitely not longer the curious student, but the in-house expert. Now, I will tell a bit more about the man beyond the science, because I think that's super interesting. Also, Professor Lesch outlined a few things there. I start off with football, because we always start off with football. We don't call it soccer, we call it football, of course. So every meeting that we have, at least the first 5 minutes are about football. We talk about Juve, Genoa, which is my favorite then. My first PhD student was from Genoa. We talk about Italy, Senegal, Africa Cup, yay. The Netherlands, and MVV, of course, and Standard Liège even. Huh? You joined me for MVV Feyenoord last year, which was not a bad match, I have to say. So football is always first. Well, let's not talk about football now, because Juve did not do so well lately. Let's talk about introductions. You are a man of introductions. You demand a proper introduction in the lab, otherwise you will not engage in a certain technology. You're very social. That was already mentioned. You organize socials within the group, you organize the pizza meeting, you take care of the coffee machine, you do almost everything. And those latter two items bring me to food and drinks. Coffee for you, just like for me, is the holy grail. He can drink cappuccino all day long, really. No, that's a joke, of course. Otherwise, he would be in trouble, I heard. And you even introduce a completely new variant of pineapple pizza to our laboratory. And then there's rum. How does the rum play a role here? Well, as part of your time in Maastricht and Hasselt, you made a deal with the great Philippos Koulesakis. We all know him. And according to persistent lab folklore, it involved reciprocal support. Yeah? You would help him with epigenetic editing. He would help you with behavioral analysis. And then I brought rum to the table as part of the deal. I think to date, you're still patiently waiting for Philippos to take care of that behavioral support part. But anyway. In the meantime, you also got engaged scientifically with Emily, which is not coincidentally one of Philippos' students from the past. And again, you're going to help each other out. Whether there's rum involved, I'm not sure yet. What else is there to say? Well, maybe missing out on so many meetings without prior notification. I'm not sure whether this is an Italian or an African issue. But yeah, you missed out on a few. So Britt, being both the paranym and partner, I think you must be a very patient person. Let me stop the roasting, because particularly Renzo this evening will take over the roasting and will continue with this. So that part I think is covered. Now, all jokes left aside, I think you're a wonderful and lovely person to hang out with, David, next to being a very talented scientist. And we're all very happy to keep you on board also as a postdoc for the coming years. And we look forward to many more pineapple pizzas and whatever meetings that bring up food and drinks, or with actually you not showing up without prior notification. Your family must be very proud. we are at least. So, congratulations. Ik heb gezegd.
Thank you.
Thank you. Dear Dr. Diouf, also on behalf of the University of Maastricht, I congratulate you with this degree you have acquired, and I will have the last word. And we met also at the Fundamental Neuroscience course in 2018 with my block, Neuroplasticity and Pain. Maybe it gives you some pain thinking about that. When reading your introduction, it was very interesting for me to see that the NCAM, neural cell adhesion molecule, pops up, because that in the past I did research on that, and now it's nice to see how the field develops in this respect. My last word is now on football, because I heard something about football, and you always begin with football, so we now will end with football. You are a fan of Juventus. I have close friends in Napoli, and so in the Italian area, I'm favorite of Napoli. But in the Netherlands, I'm fan of PSV Eindhoven. And to end, last year PSV Eindhoven beat Juventus as well as Napoli. So I would like to end with that. And with that, I close this session. Reception area, which is through this door, and then you go to the right and you will find it. After having the picture here,